My name is Ilya Nemenman. Usually I'm a professor of physics and biology at Emory University, and I'm also the director and center for theoretical biophysics and living systems. But today I'm introducing you I'm standing here as a member of the center, speaking on behalf of the center. And I would like to welcome you at our annual Hans Pagel's summer lecture. So thank you very much for coming. The center is an iconic institution in physics with the impact which is well beyond its small staff and small budget. So for example, those things such as quarks or pulsars, things that you may have heard about, they were first discussed among scientists here at the center. They're not discovered here. This is mostly a theory center. Experiments were done elsewhere, but the discussions were done here at the center. So this is a place where we brainstorm new ideas, we create, come with new ideas. For me, as Emily said, I've been coming here for 20 years. Every time when I show up, I leave with new ideas, new friendships, and these friends have been with me now for decades, right? Time flies. And I think my experience is not unique. Almost everybody who has been here once keeps on coming. And yes, hiking is important, but I think that interacting with the friends and creating new ideas and thinking quietly alone or with your friends is the most important thing. Tonight's lectures is going to be given by Leenoy Meshulam. Leenoy is a researcher at the University of Washington. She works on the problems on the intersection of neuroscience and physics. And she was previously trained in Tel Aviv University, Princeton University, MIT, and now is a researcher at the University of Washington. She holds prestigious fellowships from the Boris Wellcome Fund and the Schwartz Foundation. I'm particularly excited to introduce her because Leenoy is my academic sister. She's been trained by some of the same people that have trained me, 20 years ago. And so it's a great pleasure to welcome her here. But before I do that, before I give her the mic, I want to say a few more words about these things, right? Many of you maybe are asking or thinking about why it is that we're talking about octopuses here in the physics center, right? Isn't this biology? In high school, you take biology classes, the physics classes at a separate time, right? In fact, this is a very recent invention. Science has for a long time penetrated between, through these barriers between different disciplines. And if you go back even just 60, 70 years, the foundational Nobel Prizes in biology, such as structure of DNA, figuring out how neurons talk to each other inside of our brains, or proving that bacteria evolved in a Darwinian fashion, these were all done by teams of biologists and physicists together, right? And so in some sense, what we're doing here now in this last few decades is reclaiming the physicists as people who contribute to understanding this beautiful world around us with just a different set of methods. And so the talk today is not biology talk, it's actually a physics talk about physics phenomena for which an octopus is just a beautiful implementation or a beautiful example of. And with that, I think Leenoy is going to say a lot more about that. And so with that, let's just welcome Leenoy.
Welcome. Thank you, Ilya, for this beautiful introduction. My name is Leenoy Meshulam, and I'm incredibly excited to be a visitor to the Aspen Center of Physics this summer. My field of research is the physics of life, or as we sometimes call it, biological physics. What I would love to do today with you is to share with you where is the frontier of the field, specifically in the areas that I focused on, maybe also a few of the discoveries that we hope that the future holds. So today, we're going to talk about emergence. What is emergence? Why is it that we need to look at phenomena across scale in order to understand emergence? Then we're going to talk about emergence in living systems. I'm going to share with you my two favorite ones and the ones that I am working on. So let's talk about water. It's floating. If I were to ask you, actually, let me ask, why is the ice floating on the water? But I'm limiting your answers to one-word answer. I'm curious as to the words you would like to give.
Density.
Density. Tetrahedrons.
Sure.
Words?
Air.
Air. Actually, there's at least one more.
Phase.
Phase. Difference between the ice and the water. So you've given me phase, density, temperature, right? The difference between the ice and the water. Okay. If I didn't limit your answers to one word, ones we talked about, phase, the density, the temperature, you might have said something that sounds a little different, right? You might have said something that sounds more like in the water, Leenoy, the hydrogen bonds between the water molecules are allowing the molecules to come closer together. In the ice, the water molecules form a crystal structure. As a result of this, there is more space between the molecules. The hydrogen bonds become permanent. This is why the ice is floating. It is more rigid. It is lighter. This is great. Look at these two descriptions. Maybe the first thing we're seeing is these are a lot more compact. That is fine. The other thing is these are emergent properties of the water. Emergent properties, meaning properties that arise from the way in which the water molecules interact with one another. The way in which they interact is what dictates a special kind of organization. And this gives rise to the emergent properties, the density, to the temperature. This is not too dissimilar from what happens in this magnet we're also very familiar with. The magnet is made of many atoms. Inside the atoms, there are electrons. These electrons have spins. These spins, if I were arbitrarily, the spins are together, going all kinds of directions in the material, that's like any other material. It is not a magnet. If I am if they get organized in a very special organization, they all orient in the same direction, then a magnet emerges. And things can interact in special ways, not just in space, also time. So, for example, individual components, interactions, emergent properties. I've always thought that Debussy makes wonderful emergent properties of music. It is Clair de Lune, and you can hear the harmony, the melodies. If I were to rearrange the way in which things interact, I'm about to get very different emergent properties. Doesn't sound like Impressionism anymore. Doesn't sound like Debussy anymore, right? Can. All kinds of emergent properties. Note here is about emergence in time, and the interactions between them are actually happening in your perception. The notes themselves not interacting in the piano. It's about a special way in which they are organized inside your perception, through your ears, in your brain. So I would like to take just one moment to talk about things that might be very close to our heart, just as equally beautiful physics, are actually not emergent properties. Not emergent properties, despite the fact that they have many constituents. Gravity is a good example, something that isn't an emergent property. Same apple is going to fall just as fast. It's going to feel just the same gravity. If I were to take the Earth, someone else were to take the Earth and rearrange the way in which the parts interact. Let's say I put Australia and Africa together, Europe to a few parts, split the states around, move the oceans. The way in which things interact can change. The Earth might look a little bit less like Earth, but the apple is going to feel the same apple. It's going to feel the same amount of gravity when it falls. So this is very different than it was happening right here with the ice and the water. The ice and the water, something qualitatively different is happening at the large scale in these systems, right? If I were to examine each one of the water molecules at the small scale, in isolation, I wouldn't know, I wouldn't be able to understand the difference in the density because it is not inherent to the individual components. What it is a property of the collective behavior of many, many water molecules together. A lot of what we do in physics is try and understand emergent properties. As we do this, we've learned that scale is crucial. We need to think what happens through the scales. Sometimes we can't look at things at the small scale to understand them. We need to look at them at the large scale if we want to look at the properties that emerge. So another large-scale phenomenon that we're all very attached to and involves water molecules is the weather. Now, this also, matter of scale, is incredibly important. If I were to put sensors just around my house, what I care about, focus on the rain that's currently coming down. Really what I want is to know, is this cloud moving anytime soon? This is what I'm going to get from that. However, if I am interested in what's happening in the continent level, I might care how the hurricane that was the result of the cause of this cloud is moving and how the tornado that is happening is going around. And of course, we have global patterns of weather, like the Earth keeps getting warm over the years. So new phenomena, different phenomena, rises at every scale. If we want to look at the emergent properties of a system, we need to look across the scale, that we can see what happens. Need to look across the scales. We're interested in emergent properties. Concept, emergence, it's not new. It's all the way back to Aristotle. I'm very fond of this particular quote from the 8th book in metaphysics. Aristotle thought about this in a little bit, actually in a very similar way, very fond of the way he said this. So let me read this quote out to you. In the case of all things which have several apart, in which the totality is not, as it were, a mere heap. The whole is something besides the part. The whole is something besides the parts. There is a cause. For even in bodies, contact is the cause of unity in some cases. In others, viscosity or some other quality. We also have discussion of this concept in more current days, with the most seminal paper in the field that I highly encourage you to read. Beautiful read. Definitely changed the way I think about things. It's from Phil Anderson. It's called More is Different. In 5 years before Phil Anderson went to win the Nobel Prize. Something different, but this paper is still wonderful. And he's talking exactly about the thing we just saw with the weather. At each level of complexity, entirely new properties appear. Complexity, and at each level of it, entirely new properties appear. Okay, let's talk about life. In the living world, we have beautiful, beautiful examples of emergent phenomena. Animals self-organize themselves to beautiful shapes. Birds flock together, create these beautiful shapes in the sky. Fish seek protection in large numbers and create beautiful schools, sometimes in these spheres. Termites build incredibly intricate homes, towers of structure. It has so much complexity. Ants self-organize themselves without any leader that's telling them what to do, in literal bridges so that they can hop between spaces. For me, most astonishing emergent properties in this universe is life itself. Life may be the ultimate example of complexity, so much complexity that gives rise to this emergent phenomenon to life. Inside your body, 20,000 genes, 100,000 different proteins, 36 trillion cells. In fact, I recently learned that if you take just your immune system, that's 2 trillion cells. Means, think, right, that if I am to weigh this, it would be more than 1 kilogram. Just your immune system, 2 trillion cells. 60,000 miles of blood vessels is more than twice around the Earth. 600 muscles, 24 of them just in your arm. Together, right, each one of these components is small, maybe less sophisticated, definitely not alive. They organize together in very special ways that have to do with the way they interact to give much more sophisticated structures for heart, brain, or lungs. And together, on this scale, they interact and they organize in a very special way. Life emerges. And so as we're thinking about studying this phenomenology, right, studying emergence and studying this in living system, and I'm thinking, what system should I choose? It is everywhere in the living world. How do I think about this? What do I need in order to actually be able to study that? Maybe what I want is, given that I know that I am going to need to look at things across scales, I want access to these scales. Right? I want to maybe be able to look at all the individual components and see what's happening there. And I also want to be able to know what is the emergent properties. What are the emergent properties and what is the emergent property specifically that I'm studying that they're trying to achieve? And I want to be able to see that and monitor that and study that. So let me share with you the first model system that I'm working on. I think has these properties. Tell me if you think you know what this system is trying to achieve its emerging property. Property.
Camouflage.
Camouflage. Octopuses are artists of camouflage. They've been doing this for a very long time, and they are excellent at it. They're excellent at this because this is their main strategy for catching any prey, right, as a predator. Like many other invertebrates, their entire rigidity has to do with they have a hydrostatic skeleton. It's only fluid pressure. It cannot get more soft. Their only protection in this universe is to be able to hide really well. Their only strategy for catching prey effectively is to be able to hide really well. And they have been doing this and excelling this for a very, very, very long time. This movie was a little fast. Let's see it again. Let me tell you that this movie was taken by Keisho Osada in the southeast tip of Japan, island that's called Okinawa, off its shores. You're meeting here, in just a moment, Octopus cyanea. These are octopuses that are very common if you go diving the island of Okinawa. About this big. Fantastic camouflage. They are big, and we would really like to know what is happening on their skin, right? Let's look at them again. Very good at changing color and changing texture. Changing color, changing texture, sometimes changing its mind. They're pretty big. We would like to study them in the lab so that we can measure them really well. My experimental collaborators in Okinawa, working with a different octopus that is a lot smaller. It's about 5 centimeters big. This is Octopus laqueus, the animal we're actually working with. Maybe 10 centimeters with the arms. Octopus anatomy for a moment. This is the mental, so that's the body. The brain is under the eyes. The arms actually come out of the head. Just so they were clear, this thing had feet. It would be here. This is what it looks like in the lab. From the top, in the aquarium. We are interested in how they are camouflaging. And before we look at this, maybe you want to know something about how close are they to us, because they look very alien-y, right? Last time you and the octopus were the same thing, 600 million years ago. That is long enough ago. It is around the time that Earth moved from having single-cell organisms on it to multicellular organisms living in it. Dates that far back, the split between us and the octopuses. The octopuses are part of a family that's called cephalopods. Fish and the squid are also part of them. In that family, the skin can change color, can change texture. So the octopus skin is covered, it's tiny octopus, by no less than 100,000 pigment cells. These are skin cells. We call them chromatophores because they're like little color sacs. Every chromatophore is one color. And under each one of these chromatophores, there is a muscle that is expanding and contracting to create more surface area of this color or less. This is what it looks like, right? Different sizes and the different colors. I'm going to show you right now a movie of the skin so that you can see as the muscles are fluttering how the color and the color patches appear on the skin. Skin of cephalopod up close. We, however, are interested not just in what is happening on in the individual chromatophores, right? He said, would like to see what's happening at the different scales. We want to look at the big visual patterns that are happening on their skin. Let me show you the entire octopus. We're interested in the global visual patterns, right? What I'm about to show you is how these visual patterns play on the skin of the octopus as it transitions from one pattern to another pattern. Make a side comment here. This video is actually of a sleeping octopus in the lab. There's a little flower pot. These are the arms. That's the body. These are the eyes. This is a sleeping octopus because this video is taken from a different study of ours that I'm not going to talk about today, where we examined the sleep of octopuses. And we found that they actually change color and texture in their sleep. They play patterns that happen while they're awake, a little bit like what is happening to you in rapid eye movement, age of sleep. Right now, what we're going to focus on here is look at the transitions between the different patterns, right? To count the patterns, see how the texture changes. So we have multiple textures and color changes, and the octopus is smoothly transitioning between them. And you can see how intricate the texture and the color can get. They're changing in 3D, right? It can get spiky, what you're seeing right here, because they need to mimic a coral. They live in a coral reef. So the first thing that we realized in the system and have understood is just how hard the octopus has to work. Right? We can think about cameras from back in the day. If I have material that is sensitive to light and pinhole, this is all I need. Pixel perfect picture. This is not what's happening to the octopus. The octopus needs to become invisible, which is a little bit different than becoming exactly the same, right? I need my boundaries, be very careful with them. I need to make sure that I'm getting the texture right in the local scale, right? In the tiny scales. And I also need to make sure I'm getting this globally right. The way in which the octopus does that is it needs to look at the pattern, right, at the coral, receive that, goes through the internal signals, and present this on the skin through muscle movements. It's incredibly hard. And there are 100,000 of these muscles to play with. Importantly, octopus is not water. Well, maybe mostly water, but very important ways, it is not at all like water because the chromatophores can't allow themselves just look around locally, like the water molecules, be only interested in what's happening to my neighbor. There are symmetries in the corals. It's the way in which the light breaks. I have to be very concerned with what is happening to the chromatophore on the other side of the octopus. Otherwise, entire magic is gone. The system that is in charge of controlling all this intricacy, making sure that everything across the scale of this visual pattern works just right, is the nervous system. Nervous system, the one that is controlling everything that you're seeing on the skin. And how does the nervous system is able to execute this? 100,000 chromatophores to create exactly what's needed in a fraction of a second. Octopus does this so fast, is exactly the research we're doing right now. So now you know what keeps me up at night. What maybe you would really like to do is to peer into the thoughts of the octopus, right? To peer into the nervous system and to see how this gets coordinated. How does that happen? There is a system in which I am able to peer into thoughts, or at least to look the neural activity. And that's the mouse of a brain, in the brain of a mouse. Before I'm talking to you about the brain of a mouse, let's talk about the brain for just a moment. Tell me about the brain. Take answers. What is the most interesting to you? Yes. Without blood, it's gray. Yes. Yes.
Sentient.
Sentient, okay.
Self-aware.
Self-aware. You think the brain is self-aware and sentient? Think you are self-aware and sentient? Think this is where it's localized? Yes, maybe all three.
Plasticity.
Sorry?
Plasticity.
Plasticity. Things change over time. So a few facts so that we're all on the same page. Neurons that are the cells inside the brain, this is just an illustration, communicate with one another by electric pulses. They spike, electric spikes. The mouse has in its brain 70 million neurons. Just for comparison, your brains, 100 billion neurons. 100 billion is about as many as stars in the galaxy. It's a lot to coordinate, right? They are incredibly interconnected, the neurons, with one another, and they are what determines all of the function, all of the behavior, all of the thoughts, all of cognition, right? Largely, as a field, we actually do not understand right now how that happens, how from patterns of neural activity, from these spikes, function rises, function emerges. How is it that I can speak to you, I can throw a ball, I can have a thought? We don't know how to understand the neural patterns of activity that are happening in the brain. But we can look at them. We can look at them in in a large numbers, which is a huge stride. It happened very recently in the field. Suddenly we can look not just at 1 or 2 neurons at a time, but thousands of them at a time. There are 2,000 neurons inside a breathing, moving, living mouse. The area in which we're looking at is the hippocampus. This is an area that's thought to be crucial for memories. Each one of the little donut ones here is a neuron. And the reason that you can see them by eye, this is under the microscope, is because neuroscientists have figured out how to genetically program the neurons flash light every time they spike, every time they get active. So we can see the flashing lights is their activity, which I'm going to show you in a moment. See the light? I don't know if you can see the movie well. See the light? Really? I don't know if... Thank you. Right. First thing to notice, neurons are not all active together. That's good. When they are, that's a disease stage. Second thing to notice, also not active just one at a time. Few of them are active together, others come up and get active, then others come up and get active. These patterns are incredibly complex, right? Incredibly complex. They are controlling lots and lots of things. Remember, with the neurons, it's not even the complexity of I have multiple colors and I need to be aware of the chromatophore that's far away from me. It's even more, right? 100 billion of them for you, 70 million of them from the mouse. They need to talk to neurons that are very far away from each other. I need to be able to coordinate my movements with the visual input that is coming in, with my auditory output as I'm speaking to you. Everything very, very, very fast. And neurons don't talk just to the ones next to them, also not just to a few that are far away. One neuron can talk to 100 other neurons, to 1,000 other neurons. They can do this between totally different brain areas. So the complexity here is enormous, right? Enormous. And so as we're looking at this incredibly complex system, what are we going to do? I am not deterred by complexity. You know why I'm not deterred by complexity? Why all of this is okay? Happily, I'm a physicist. I'm a theoretical physicist, which means that I took none of the measurements that you saw here. We'll talk at the end about the people who did. My job is to write theoretical frameworks, mathematical frameworks, to understand the measurements that they are taking. It's happy that I'm a theoretical physicist, not just because it's wonderful company, but also because I know what to do when there's complexity. I've seen complexity. We've all seen complexity. You're all with me being theoretical physicists right now. Water is incredibly complex. In fact, right now in physics, in fluid dynamics, the most we can do is simulate 120 water molecules. With the biggest and best of computers, we can't get water. We have to describe this based on its emergent properties. So I know what to do with complexity. Now, granted, we have more complexity in the brain, the octopus, only in the brain where the neurons speak to ones that are far and wide and where they need to coordinate so much and so fast. But I know where to start, right? At least where to start. And I also know what seems to be important when I'm talking about the emergent phenomena is the scales. So I want to be concerned with what's happening in the larger scales, which means that I want to examine my nervous system, the brain, across scales. I'm going to do this. Talked about the fact that the neurons can talk very far away, communicate with other neurons that are very far away from them, which means that maybe the way in which I want to look across scales is not just this neuron, now the 5 that are around it, now the 10 that are around it. I am looking at groups of neurons, which is what I'm going to do. I am going to group them by the similarity of their activity, which means that if my neuron here is very, very similar in activity to the neuron here, I'm going to group those together. And I'm going to build small groups. Then I'm going to make bigger groups out of those groups and even bigger groups out of those groups. And I'm going to look at what happens and measure quantities across these groups. Look across these scales. I have 2,000 of those. You can start in twos, in couples, go to 4, go to 200. See what happens. See what happens. This is what happens. Before I am explaining to you exactly this plot, I am going to claim to you right now, and I'm going to explain in a moment, that this means there is scale invariance in the system. And for you to be as astonished as me at this beautiful straight line, I would like to have a few minutes left, deep dive for just one moment into a little bit of math so that you can share the excitement with me. Bear with me for just a moment. What is scale invariance? Scale invariance means that as I am zooming in or I am zooming out, things stay exactly the same. The pattern stays the same. This is the Mandelbrot set. It is here, the visual aid for you to understand what I'm saying. I'm saying patterns that stay the same no matter how much I'm zooming in or how much I'm zooming out. And there is a function, mathematical function, you have all heard of before. Is just that one that describes scale invariance. When things are scale invariant, what I have is a power law. This is a power law. And what you'll be able to see in this curve is that no matter how much I'm zooming in, which I'm going to do in a moment, or zooming out, this curve is going to look exactly the same. So follow the curve. We're going to zoom out. Exactly the same. Zooming in, zooming out, scale invariance. When I see a power law, it means I have scale invariance. You're seeing how all these points here are very dense, and they get farther and farther away from me. There is a fun trick that we can do to visualize power law better, which is we stretch the axes in a particular way, form a logarithm. We make it such that they are no longer evenly spaced. and then see what happens to this. What's going to happen is it's going to become a straight line. And I present it this way. So let's look at this. I'm going to stretch my axis in a particular way from a logarithm. See, it became a straight line and back. I'm going to do this again, and you're going to look for me the x-axis, the y-axis. You're going to see that the decades, 10, 100, 1,000, now are actually evenly spaced. We can see it much larger ranges. So when I see a power law, any kind of calculation that I did gave me a power law, the data. Then I know it's scale invariant. Things look exactly the same as I'm zooming out or zooming in. Exactly the same across the scales. Let's look again about our results. You see the x-axis. Decades are evenly spaced. These are not, because we stretch them from the logarithm. And I'm seeing the straight line. Drew the line for you, these dots from the data. This is taken from calculation on real neurons, analysis on real neurons. They were so messy and so complicated. Look at this beautiful line. Things look the same across scales. In fact, we saw this particular straight line is from the variance of the activity as I'm looking at these groups. We saw this in many other properties. We saw this in how much silence they are, in their modes, in their density of activity, in so many other things. In all of them, as we were grouping neurons together, looking at the quantity, we were seeing that it looks the same. I'm zooming in, we're zooming out. The small groups, the large groups. We have found evidence of scale invariance in the brain. Now, parenthesis here, this is a particular region of the brain of the mouse. People have replicated these results since in other brain regions of the mouse, in other animals. It's still worthwhile to be very careful with conclusions. From what it seems like right now, instead of finding, right, like in my weather example, that in every level, new property appears, my magic is everywhere, right? Instead of finding that, I don't know, maybe at the level of the 2,000 neurons that things emerge, something really special happened that didn't happen before, I actually can get there, right? And we got to that, we got to this idea that we need to look across scales and go here, following the same logic that followed us for the magnet, for the water. We knew from that that we need to look across the scales, right? We knew from that not to be alarmed by the complexity and think, oh, maybe the detailed under are something that I can decide for just one moment. Look at the larger scales and see what's happening there if I want to examine emergent properties, which are my heart's desire here, right? The function, the thoughts, throwing a ball. So it could have easily been the situation. What is the alternative here? It could have easily been the situation that because of all the added complexity of the brain, right? Neurons are far away from each other. They're very heterogeneous. There are so many cell types of neurons, so many cell types, different ones from one another. They communicate with faraway neurons. It could have been the situation that I will discover nothing, right? And going across scales won't help me because maybe there is a different rule and a different phenomenology that happens in every corner to every cell type at every scale. It can have so many of those. The incredible complexity. Instead, find simplicity in this incredibly complex system. Find simplicity, which is to say, there are only a few rules that I just need to follow the entire time to get from the small scale to the large scale. That means, we think this might make sense, right? Because thinking about the brain, I need brain that gets organized and gets developed from a different rule in every corner. It's very difficult to implement. These are 100 billion neurons, and that's not even the most common cell in brain. We have glias, and we have blood cells, and we have like so many other things. We need to organize all this. Neurons are the ones communicating, giving rise to all this behavior. Brain needs to follow a few rules of organization, principles of organization. So they are common to everything. That's a lot simpler. And on the scientist side, or researching this, it is highly encouraging. It's highly encouraging because if there are only a few rules I need to understand, then maybe I have a chance of doing that. Maybe we're a lot closer to actually figuring them out. And once we figure those out, that is the heart of the secret. It is the heart of how thoughts and action and behavior emerge from all of this complexity, from 100 billion neurons in your brains. I understand that. I can intervene, I can correct, I can understand how we are able to think different things, how we're able to throw the balls. I'm understanding the brain. Thank you. So everything that I showed you today is a result of a collaboration. Specifically, I'm a theorist. I'm totally dependent on wonderful experimental collaborators. We're working together. I'm very fortunate to have, I think, the best collaborators in the world. So for the project of the mouse, these three, this is Bill Bialik. He's also on the theory side from Princeton University, involving on the scale invariance results found in the mouse. David Tank and Jeff Gauthier are on the experiment side, the beautiful videos of the brain. Sam Ryder runs a lab in Okinawa Institute of Technology in Japan. It's a lab that researches octopuses and squid and all the cephalopods. DT is the one who was looking at the octopuses inside the lab. Omi is the one who's trying to record them, and Keishu is the one who dives underwater to produce these beautiful videos. I'm incredibly grateful for all of them. Yes.
I have two questions. Have you characterized any of the gene circuits or expression systems, specifically structure and functions, with help from like maybe the Institute of Protein Design?
Did you hear the question? Sorry, you finished?
Yeah, and then also any connections with the neural networks and convolution models?
Let me do one at a time. So the first one was if we characterize any of the genes that are involved in these processes, right? Maybe an answer in two parts. One, I couldn't possibly do this even if I wanted, because I actually don't run experiments. But neuroscience as a field has characterized many of the cell types that are happening. There is transcriptomics, the Allen Institute of Brain Science that's located in Seattle, has actually characterized cell types. And they find interesting results, for example, that types are actually not quite a discrete thing as you think. It's actually kind of a continuum. And so people are definitely working on it and getting kind of maps the entire brain for all of these things, working on characterizing. The other question, remind me?
Convolution models in connection with
so this is a question about artificial neural networks. We're not mentioned here at all. We're talking about deep neural networks, one that have, these are simulated networks, which we can have particular functions that they perform. Particular question is about when you put together, when you put them together with a function that's called a convolution. So this is a particular kind of multiplication. What happens through the layers of this deep network? Let me say maybe the first thing, which is that deep neural networks that are incredibly strong in processing many things, can perform incredible computations, work very differently than the brain. One of the mysteries in what's happening in the brain is that there is a lot of recurrency. There's a lot of feedback, a lot of loops. Part of the power of convolutional neural networks, as we have found recently in machine learning, is that they are completely feed forward. The connections go only one direction, from the first layer to the second to the third, all the way up. And so that way, they're limited in ways that the brain is not. So they're artificial. The brain has more going for it. On the other hand, artificial networks have started from a completely different place because the brain cannot just stop functioning until it performs the function. It's not starting from nothing with evolutionary constraints. So these systems are relatively different. Maybe a comment to say about how they are similar, which is that people have managed to execute, to implement things like visual system in deep neural nets and see that some of the properties of the artificial cells share similarities with things that are happening in the visual cortex. Yes.
The description you had of the neurons talking to each other sounded remotely like quantum entanglement. Is there any relationship?
Okay. Something I'm going to say and something I'm never going to say about this at all, ever. First thing that I am going to say is maybe clarify the notion of neural communication. Which is to say, I was talking about talking to each other, really what is happening is neurons are a few tens of microns long, right? They have processes, they have arms that extends from them, axons, dendrites. There is a little bit of a gap between the end of the arm of one neuron and the next arm, the next neuron. So there is an electric signal that goes down the axon. It gets transformed into molecules that jump this gap. And then the other side, electric pulse that again into the next neuron. So that is the type of communication. As I describe this, its classical format, as we understand it, has nothing to do with quantum entanglement. There is a subset of people who are interested in both these topics that are looking at some of what's happening in neurons in relation to quantum. I think it's very much still in development, so I'm actually not going to comment on that particular thing. Yes?
I was just curious, you said the mouse was living and breathing. What was the mouse doing when you were the neurons in the brain of the mouse that were firing?
Thank you for asking this. I didn't get to brag the experimental collaboration and just how wonderful they are. Jeff Gauthier that I mentioned, the question was, mentioned that when we're looking at the brain of the mouse was behaving and alive. And what was the mouse doing? I forgot to say the mouse was running. I didn't tell you where was it running. What Jeff does for a living is play video games with mice. So this is a setup, virtual reality. Okay, there is a ball, mouse is on the ball. The head of the mouse is fixed because they need to put a really big, fantastic microscope above it, 2-photon microscope with a resonance scanner. So you need the mouse to be, the head to be very still, but the body can move. So they figured out, okay, let's just fix the head. Body can move on a ball. It's a styrofoam ball and the 270 degrees of a screen, like an IMAX for mice, that the world is projected on. And this mouse is running down a virtual corridor. In this virtual corridor, they can give it all kinds of visual cues. There can be grapes on the side, can be, kind of triangles on the other side. You can make decisions. Am I going right? Am I going left? It's going to turn the ball, go right, go left. Head is still fixed. We can still look at the brain. The mouse is moving in the virtual world. Thank you. Yes.
What is the role of nonlinearity? Could emergent phenomena arise in a linear system?
Okay. Question was, what is the role of nonlinearity, and can emergent phenomena arise in linear systems? I think that question has particular mathematical implementations, which we can talk about. But overall, I want to stress something else, which is to say that most of the time that we're talking about emergent phenomena, we're talking about a huge number of particles, huge number of components. As we're thinking about the way they interact, it's not quite about whether this is linear or not. It's about how is the special organization happening. So I don't know if one can construct a system that is linear. Most of the world is nonlinear in what it is doing. But I don't know that I have a direct answer to the entire system and whether it is linear or not. But I am going to say it is not a property of any math that is happening there about linearity or nonlinearity. It is a property of the way they interact in the special organization. So in that sense, it's a little bit of a different way to think about that. When we can write down something that is linear or something that is nonlinear and is very concrete, is already a huge progress for us in complex systems, because the problem is we can't even write this down. Yes?
How do they talk to neurons that aren't right next to them? Describe this like two next.
Yeah, so these arms get very, very, very long. In a giraffe, it's like the entire neck. You can get very long. These processes neurons can get very long, but most of the time, it's actually not just the length of the arm. Most of the time, what's happening is that they actually are moving the signal away. So there will be a lot of neurons on the way to interacting with a neuron that is far away. It's very fast, and it is very accurate because they're losing very little the energy of the signal on the way. So they're actually effectively interacting very far away. Yes.
Like the game of telephone where A talks to B.
But a good game of telephone.
A to B and B to C and whatever?
Yes.
As opposed to A to Z?
Yes, most of the time it's A to B and B to C. Sometimes A to B, C, D, E, and F. B, Z, Y, X, and A to, all together because they can have many arms. They're not limited like me. Yes. Yes, thank you. Leenoy life history. I actually was working in the mice before, a system that's very close to my heart, because I wanted to look at something that's really large scale and we can see the interactions because I'm fascinated by emergence. But 2,000 neurons is 2,000 neurons out of 70 million neurons in the mouse. And while it's only very recently that we can even see a few thousands, it is still only few thousands. I was thinking, after we saw the scale invariance and what is happening, wouldn't it be nice if I could look at all of my individual components at the small scale, right? Really go all the way from the first scale to the largest scale. What system do we have this in? So it has to be smaller, I have to be able to look at all these things. I want something that does something very complex, right? I want something that's emergent. So very small systems barely have anything emergent that we can do this in, maybe less interesting. I want something that has lots and lots of configurations, the microscopic scale, and giving me the same thing at the macroscopic scale so that I can research that. I was thinking about this, the paper, the group of Gilles Laurent in Germany, Sam, who is currently my collaborator, the first author of when he was a postdoc. It was a paper about cuttlefish and how chromatophores work. The important sentence that I read there was that in camouflage for the chromatophores, the first time they were able to get animals to camouflage in the lab, the system happened, there is one-to-one relationship between the neuron that is controlling the muscle and the chromatophore. Like 2-3 neurons or 2-3 chromatophores, which effectively means that I don't need the neurons in the octopus. I can just look at the chromatophores, and I don't even need to reprogram them to flash light because they are already colorful. So if I'm seeing all 100,000 of them, I am seeing the entire, right, the entire control of the nervous system of them. And I thought, wow, that seems easier and really nice to be able to look at. I also know the another advantage of the system is that I know what they need to do, right? The mouse brain, their thoughts, their actions, there are a lot of things that are happening. It's a lot closer to us. In the skin of an octopus, right? Because it's not their brain, I know what they need to achieve. I know what is doing really well as an emergent property. It will look like the coral. I know what he's not like. No octopus. Doesn't look like a coral. This is dangerous. So I have access to all the microscopic components. I know what is the emergent property. I can compare this to what's happening in the mouse, where we have a different set of tools. What's happening in the fly, what's happening in other systems that I'm working on.
I think we're going to take one last question and then we'll.
I'm going to ask you about further applications for this research and what you're most excited about.
I would really like to figure this out. It really keeps me up at night. Like the octopus is, okay, I don't know how this works, right? You have, it's so complicated. So 100,000 chromatophores, you can see all of them. I still don't know how this works. We need to be able to understand how this gets coordinated. So I need to develop a mathematical framework of seeing what happens across the scales, starting to see that maybe there's some invariance there as well. How does this get controlled? How is it through the nervous system? This is what I'm really, really excited about, understanding how the camouflage is actually getting controlled. I'm also very excited about seeing how far the scale invariance idea goes in the mouse, because while it is encouraging that it happens to us in 2,000 neurons, if I had 2 million neurons, right, be able to see if this keeps going, and then maybe I'll understand what are the few rules, right? When I'm talking about temperature here, I can actually get there from the individual water molecules. It's a guess about the property. I need to know how this looks at the large scale to be able to guess how that works. I don't know what are the units of thoughts. I would like to know the units. Thank you.